This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on October 20th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today, I'm on the campus of the National Institutes of Health, and my guest, who I've been wanting to speak with a long time, is Jeffery Taubenberger. Welcome to TWiV.
Thank you, Vincent. I appreciate the invitation.
My pleasure. I'm here for ID Week, so I thought I would try and see if you were available. So I'm glad it worked out.
Yeah, me too. It's great to chat.
And I just wanted to mention, in case Brett is listening, Brett Jagger, right? Or Jagger?
Jagger.
Like Mick Jagger, right?
Yeah.
I met him at a restaurant yesterday, and we had never met, and he recognized me because, like everyone else, TWiV has gotten pretty wide, so we're recognized among scientists. And I told him I was coming to talk to you, and he told me he trained with you.
He did
years ago.
Yeah. Yeah, he did his MD PhD, did his PhD in the NIH Oxford Cambridge program. So I was the NIH mentor, and Paul Degard in the UK was his Cambridge mentor.
Excellent, excellent. All right, so I wanted to talk with you about 1918 influenza. And I think it's timely that people coming out of a big pandemic, we talk about what we learned from that. And I think most of our listeners don't know much about it. So let's tackle that. But I wanted to first understand how you got interested in influenza.
It's a kind of a crazy story, how I got interested in influenza. So let me back up a little bit. I did an MD PhD, and my...
Where was that?
In the Medical College of Virginia in Richmond, Virginia. And my research interest was hematopoietic stem cell development, specifically looking at how hematopoietic stem cells from the bone marrow would commit to the lymphocyte lineage to become T cells through the thymus or to become B cells. I ended up doing a pathology residency, and I did that here at NIH through the Cancer Institute, and then stayed on as a staff pathologist and continued my research on stem cell development. And then I got recruited in 1993 to go to the Armed Forces Institute of Pathology, or AFIP, on the Walter Reed campus at the time, with a new, very exciting opportunity, which was to use brand new molecular biology techniques in the service of diagnostic pathology, so that rather than looking at stuff under the microscope, we would be able to make diagnoses of looking for specific translocations in leukemias and lymphomas and sarcomas, or for infectious disease diagnosis, by PCR and other mechanisms. But the key was to try to figure out how to do molecular analysis from formalin-fixed, paraffin-embedded tissues, which is the way pathology is processed. So, back then, the general assessment was that formalin-fixed tissue would not yield any nucleic acids, either DNA nor RNA, especially not RNA. But it turns out that if you look for really tiny fragments, they are there, and you can do useful analyses. And so we started doing that, and I'm trying to think of what we could do to show why this was useful. And so a crazy idea that happened around 1995 or so was to look for tissues of soldiers who were infected with and then subsequently died of the influenza pandemic in 1918, whose autopsy tissues and clinical records were still maintained at the AFIP archives. And so it seemed like a completely crazy idea. I had never worked on influenza before. I had been doing some other kind of viral work. Just before that, we had done a lot of work on characterizing novel morbilliviruses in dolphins and other small cetaceans. And that's what led me to think that even though tissues were poorly preserved, or were either poorly preserved and then subsequently fixed, it's still possible to do some molecular analysis. So we were able to show that there were distinguishable morbilliviruses in these dolphin populations and these big die-offs that were occurring. So that is what led me to think about the influenza in 1918. And, and I was completely ignorant. I remembered one sentence in one lecture in med school where somebody said 1918 flu really bad, millions of people died. That was the extent of what I remembered, right? That was my fund of knowledge when we started this project. So, I started reading the literature. Books, Alfred Crosby's book, America's Forgotten Pandemic, really great read. This was decades before John Barry's book. And as you know, anyone who reads about the 1918 flu or learns about it, it's unbelievably fascinating. I mean, in a horrible sense. I mean, in a train wreck sense. I mean, tens of millions of people died. It had an enormous impact on the entire world. Probably, resulted in finally coming up with an excuse to end World War I. It really had an enormous impact. And so the more you got into it, the more determined I became that we should try to do something about it, because here was a huge pandemic, probably the worst pandemic that has happened in recorded history, for which at that point we really knew very little, right? We knew nothing about the pathogen itself. It seemed likely that it was an influenza A virus, but there was no isolate. There was no way to understand how it formed, why it behaved the way it did. So that was the idea. That was the kind of simplistic idea.
And this is 1995-ish.
Yeah, 1995. Yeah, so this is, almost 30 years ago now. I'm looking back on this, and, I think there's something valuable to being so young in science. I think Sidney Brenner had some kind of quote, I can't get it quite right, but, about ignorance is a really important factor in doing science, and that once you're in a field for a long time, you learn all the things that are impossible, and I didn't know that what I wanted to do was impossible, right, so we did it, right. And so I had very talented molecular biologists with me in the lab, Anne Reed, who also became very passionate about this project and, played a key role.
There's a line from Alice in Wonderland from the Queen. I've thought 7 impossible things before breakfast.
Right.
You have to dream, right?
Yes.
And not be limited. But you're right. As you get older, you say, that won't work.
That won't work. Yeah. Yeah. No, I think, that's probably why, a lot of scientific things, happen when people are in their 30s and so on. Anyway.
So that was done before you came to NIH?
Oh, yeah. So I didn't come to NIH until 2006. So the whole project to find and sequence the genome of the 1918 virus, and ultimately its reconstruction in this collaborative effort, which I'm sure we can talk about, all happened prior to my coming to NIH. And then in 2006, it became possible for me to move here to the intramural program of NIAID to set up an influenza pathogenesis lab with nice high containment facilities where I could continue to work on flu pathogenesis and et cetera.
Okay. Let me take a pause and just check my camera to be sure before we go on. Okay. So all this happened long after I left Peter Palesi's lab. You may know I did my PhD with him.
Do know.
I was his first PhD student, actually. Oh my gosh. I never got so much attention from someone. I was his only person at the time. And every day he would talk to me. This was 1975 to 79.
Okay. Mm-hmm.
And there was no cloning yet. Mm-hmm. There was no sequencing. We used to do fingerprints. Remember RNA T1 fingerprints?
Yeah.
And I just remember being in his lab when 2 things happened. First was swine flu 76, and he did some fingerprints. He said, this is a pig virus. This is not a human virus. But Ed Kilburn didn't listen to him. Ed Kilburn was the chairman of that department. And then a year later, the 1977 H1N1, and he fingerprinted. He said, this looks like a 1950s virus. So those made big impressions on me. The interface of science with the public, which doesn't get much better than with, influenza viruses and now coronaviruses, of course. Although I ended up leaving, I was going to go back to flu, but then I did some interesting things with polio, and I couldn't resist staying in polio as well. So your work came many years later, and I always loved this. So I want to talk about this recovery and related questions, but what is influenza? Let's start with that. You're a doctor. You can describe influenza as a disease.
Right. So influenza as a disease has been recognized for hundreds of years. The term influenza actually was applied in the Renaissance period because there was seasonality to this, that it happened generally in the winter months. So influenza was under the influence of the stars for that part of the season in the winter months where people tended to get influenza. You look at medical records from even the 1300s and people say everyone gets influenza, but infants and the elderly and pregnant women and other other really chronically ill people are the ones that are most likely to die. So the basic picture of influenza as a very transmissible disease, obviously in a pre-infectious disease era, in probably more miasma-y era, but the clinical features of influenza are widely recognized. It's an acute infection, generally mostly upper respiratory, sometimes lower respiratory, generally self-limited and acute. But unlike a cold virus infection, it tends to be more severe even for a young, healthy person. Flu sort of can, give you a high fever and make you feel like you're sick enough to want to stay in bed for a few days, or maybe even a week, can really knock you out, make you feel lethargic for weeks and weeks to come. Flu is bad. One of my colleagues here at NIH, Greg Folkers, has a little flu algorithm that he wrote out on a blue sticky note, which I've made a slide of and I've shown in talks all the time, which says, do you have the flu, yes or no? And it's yes. Do you feel like you've been hit by a train? Then you may have the flu. No, then you do not have the flu. So under yes, have you been hit by a train? Yes or no? No. Then you do have influenza, right? So that is I think a good way to say it.
And there's enough deaths and that's why we have a vaccine.
Right. So flu, so we were talking about human clinical flu. Flu, the virus, family of virus, obviously an incredibly diverse family of viruses, and even influenza A viruses, which we're mostly concerned about, are in, dozens and dozens, or maybe even more than 100 warm-blooded animal species, birds, mammals, bats, an incredibly diverse problem. But human influenza, year in and year out, is a big killer. Tens of thousands of people die in a typical year in the United States of influenza. So vaccine control of influenza has always been something that has been thought of as a huge public health priority for the last 60 years or so, when they started programs to actually develop the initial influenza vaccines after World War II. As you know, influenza viruses rapidly mutate, and especially the surface proteins, incredibly plastic, and can undergo very significant antigenic drift very rapidly, so that the idea of making a vaccine that matches the particular strain that's circulating is a little bit difficult, and we're always chasing after the virus, not in front of it. So, we have to change the vaccine every single year, and now, we're beginning to see that kind of variability with SARS-CoV-2 as well, which, has made control of influenza through vaccination, extremely difficult.
Yeah. So what's the biggest risk factor for dying? Is it age?
That's a very difficult question. I think that there are a number of interrelated factors. I think the risk factors for severe disease, relate to host factors. So that relates to your age, your exposure history to prior influenza viruses, your underlying comorbidities, genetic variation for which we know very little about, differences in inherent virulence and pathogenicity of the virus, which vary greatly. And then your inflammatory and immune responses, and it forms this kind of overlapping Venn diagram where all those things are contributors. But on the host side, underlying factors, pre-existing cardiac disease, pulmonary disease, immunosuppression, and pregnancy probably related to immunosuppression are big factors, and age. So certainly for seasonal flu, the age group that has the highest mortality rate tend to be the elderly. Another strange thing, however, is that the longer you live, the more different flu viruses you're exposed to. And in some cases, like in 1918, it looks like the elderly had some protection, possibly from distantly circulating influenza viruses in the mid-19th century. So it's not just a simple factor of being elderly. It's a combination of all these things.
And in fact, in 2009, H1N1 emerged, and people who had survived 1918 had some protection.
Right. Anybody who had been around before 1957, when the old seasonal H1N1 virus derived right from 1918 was replaced by the new pandemic H2N2 virus, those old 1977 type H1N1 viruses also went extinct, but people who had been exposed to those pre-1957 H1 viruses definitely seem to have some, protective memory against the 2009 virus.
So is there also an inflammatory component of the disease as we see for COVID? Right.
There certainly is an inflammatory component, and I think that that's what separates kind of self-limited illness from more severe illness, especially that would result in a viral pneumonia. That in some viruses, like the 1918 virus and other avian-derived viruses that we know of, induce a really strong pro-inflammatory response, and I think that's a significant component of the disease. And that's something that people have been speculating about for decades, and we're just now beginning to understand that. We still obviously haven't done enough work to really fully characterize it, but it certainly is a big component.
Because in seasonal influenza, most of the mortality is a consequence of pneumonia, right? That's how we actually track excess deaths by pneumonia ascribed to influenza, right?
Right. So, I mean, as you know, influenza at a kind of a population level and epidemiological level, when influenza is circulating, it's sort of easy to see. It spreads really rapidly. Large numbers of people have typical influenza-like illness symptoms. But curiously, as an individual doctor, if you're sitting in a clinic or an emergency room and someone presents with an influenza-like illness, they may or not have influenza, right? And now they may have SARS-CoV-2, but they may have parainfluenza. They may have RSV, any number of other respiratory viruses. And so paradoxically, influenza is more visible in a community spread than on an individual level. But flu is, has always been an incredibly important factor. And, the morbidity, mortality, and the economic cost associated with that is, ginormous.
At Columbia, in the winter, students all have some respiratory illness. They go to the clinic, they have a flu-like illness. They don't even get tested. They just get Tamiflu.
Right.
You probably have flu. But as you say, they may have other things as well.
Right. Right. And then, of course, there are people who are, subclinically or, just asymptomatically infected.
Sure.
And so that also makes the community spread that much more complicated and difficult.
So let's talk about 1918. First, when did it start? And when did it end?
I don't know. So when did it start? I mean, let's put it this way. It was recognized in 1918. And so the huge outbreak that everyone agrees is a huge pandemic outbreak all over the world was in the September to November months of 1918, curiously, both in the northern and southern hemispheres at the same time. Now, I say that because, as we've just said, seasonal flu tends to be seasonal, mainly in the winter months, so that we have two flu seasons in the northern and southern hemisphere that are 6 months apart. But when a pandemic virus emerges, it doesn't seem to follow those rules of seasonality. It appears when it appears. And I think that the 1918 virus was circulating certainly in the spring of 1918. It's certainly possible to me that it was circulating even at low levels in the fall, winter of the 1917-1918 flu season, under the seasonal flu season. And herein lies the problem. In 1918, of course, influenza virus was recognized clinically, but the causative agent wasn't known. There were no diagnostic tests. But this confusion of, do you have flu or RSV or paraflu, right? I mean, they wouldn't have known that. So the only thing that was visible is an extremely unusual series of serious illnesses and especially unexpectedly high numbers of influenza pneumonia deaths. And if flu has a R0 spread somewhere, between 1.5 and 1 and 2, maybe at the upper limits, it takes a long time for one person to infect 1.5 people to infect 3 people,
Sure.
And so it's going to circulate under the radar for a long time. We have a sequence from a case from May 11th, 1918. From a soldier that died of a streptococcal pneumonia that still has influenza RNA in it that matches the 1918 sequence. So for certain, and I doubt very much that this person was the index case, the virus was circulating probably widely in May. And again, I think that it was at the end of the seasonal flu, whatever subtype that was or subtypes that were circulating before 1918, so say the winter season of 1917, 1918. And then probably outcompeted it right at the end in the spring. And then because flu viruses in general don't really spread well in the summer because of temperature and humidity, kids not being in school, other factors that we still don't fully understand, there wasn't much flu. And then in September, kids go back to school, it gets a little cooler, typical flu season starts, and bam, you had this huge outbreak. And it happened everywhere at once, where communication was by steamship and rail. And so the virus had to have
no
been
internet back then.
right. The virus had to have been seeded,
Sure.
everywhere. So by the time the pandemic was recognized, it was already a global pandemic. So it was bad enough in 1918, but you think about, ask the question, where and how and when did the 1957, 68, 2009, and COVID-19 pandemics emerge? So these last two, in a fully highly industrialized molecular-based surveillance system where we monitor diseases quite closely compared to and have a lot more information than we had in 1918. And we don't actually know the answer to any of those questions either. We have speculations about where did the 2009 pandemic emerge through reassortment with different influenza viruses in pigs and combinations of human, swine, and avian influenza viruses. But how did it get into people? And why did that genotype work where other reassortment genotypes of swine virus didn't work? And, how long was it circulating at a low level before it was detected? And why did it take off? I mean, these are questions that have biological explanations, but we're just so ignorant of how this whole process works that even today, we just can't address those questions. And it's really humbling.
Yeah. But even in 1918, was the word pandemic used?
Yeah.
It was?
The word pandemic, obviously words like epidemic and pandemic and things, can mean different things to different people or at different times. But certainly this idea of, a pandemo, something, from the Greek affecting all people, was thought of, and discussed, at the time of 1918. And certainly it was recognized as a quote unquote pandemic. So if we don't know where it started, I can answer your second part of your question, which is we don't know when it ended. Okay. And it could be that the answer is it hasn't. So, the 1918, the pandemic part, the part where it's a completely brand new virus, nobody has pre-existing immunity directly to this virus, it can spread efficiently human to human. There were no vaccines. There was nothing to stop it spread in 1918, 1919. So basically, everybody on Earth, except for incredibly isolated people on the Reunion Island and, some places, escaped. But everybody else on Earth got exposed to the virus. After that, influenza viruses, if they're going to survive in humans, have to undergo enough antigenic drift of their surface proteins so that they can continue to circulate by escaping pre-existing immunity, and so have become this race of ever-evolving seasonal influenza. So the seasonal flu of 20, 21, 22, up through.
I always ask, what is a pandemic? Because today, if you ask someone, no one has, WHO has a definition, other people have definitions. It's not just a lot of people infected, because rhinoviruses infect everyone all the time, and we don't call that a pandemic. But as you said, it must be the emergency part where people are being disrupted. Mm-hmm. And Paul Offit said, and I like this one, a pandemic is where everyone is affected, but not everyone is infected.
Mm-hmm.
Maybe that's good.
No, I think it's good. I think that the problem is that the term pandemic is clearly an epidemiological or a public health term. Mm-hmm. At a virologic level, it's very difficult because, like you said, there are endemic viruses that still widely circulate. So, is it endemic? Is it pandemic since everyone gets infected? I don't know. And there are seasonal virus drifts that are much more severe than others for reasons that, we have not yet fully worked out. Sometimes the H3N2 viruses kill a lot more people than other years, right?
Yeah.
And in those years, there are influenza and pneumonia deaths higher than the 1968 or 57 pandemics. And yet they're not called pandemics. Those new drift variants rapidly spread around the world and kind of take over as a novel virus. So why are they not pandemic? So it becomes very complicated. And I think that for flu, at least, since we have to deal with the continual circulation of seasonal influenza A and B viruses, but influenza A viruses of different subtypes, that we think of pandemics as a genetically novel virus that has entered the population. So I think from a virologic standpoint, that's what I would say is a better definition. A virus at least with novel surface proteins. By novel, I mean every single flu virus is novel in the sense that, it's such a high error rate, but I mean a significant genetic variation.
Okay, so clearly 1968, 1918, as we're talking about, they're all new viruses that we haven't seen before. SARS-1 was new, but it didn't really spread enough to be called a pandemic, right?
It was pretty close. I mean, it was on multiple continents. I mean, we really lucked out. I mean, I think it was an almost pandemic.
Right. Yeah. And clearly SARS-CoV-2 is, and maybe at some point where it becomes seasonal and endemic, it will no longer be a pandemic.
Right.
But I agree. I think your definition of a new virus to which there's no pre-existing immunity is good.
So a virologic definition of a pandemic or something.
Yeah, I like that it's a brand new virus and therefore it spreads unchecked. And SARS-CoV-2 obviously fits that bill. 1918 did. So I like that definition. I mean, obviously then many people are infected and there is widespread disruption as a consequence and so forth. Okay, so before 1918, did we recognize any influenza pandemics, or was that the first one?
No, certainly we recognized any number of pandemics going back at least 500 years. So this, David Morin's here, my colleague here at NIH, and I have a quirky interest in history as well as infectious diseases. And so we have done a number of historical evaluations. And, looking at the European medical literature, which is what we have most access to going back to the late Middle Ages, there certainly have been pandemics averaging about every 30 years, but in no predictable pattern since at least 1510. And there's likelihoods of flu pandemics prior to that, although the medical records get more difficult to interpret. For example, in 1173, we see in these annual reports written by monks in monasteries in England and in France and in Italy who weren't in communication with each other, all describing, a really bad seasonal respiratory virus that caused a pest, and among many people who were sick and many people died, and this kind of stuff. It was certainly likely to be a pandemic. It's hard to know how long flu viruses have been in humans, but certainly, hundreds of years, if not longer.
Well, in fact, I read a very nice review that you and David Morins wrote on the 1918. And there's a quote from that where you write, all pandemic and epidemic influenza is descended from 1918. Could you explain that?
Yeah. So back to this sort of question of when did the pandemic end? The answer is probably never, is that the 1918 virus, in a sense, has never gone away. Now, all the human viruses that we have and other bacteria, for example, are likely have come as zoonotic exposures from other animals, especially since we started agriculture whenever, 10 plus thousand years ago, MTB and other things. But I've lost my train of thought. I'm sorry. I'm going to have to do that again. What was the question?
The all pandemic and epidemic descended.
Let me try this again. Take two. So that once the 1918 virus started circulating in humans, it seems to have wiped out any influenza viruses that were circulating prior to that, which other pandemics have done.
Right.
But in the case of 1918, it was the direct the direct descendants of 1918 were human seasonal H1N1 viruses and classical swine H1N1 viruses that then adapted to those two different species and have been evolving separately since 1918. That in 1957, we got a new pandemic in which 3 of the 8 gene segments were replaced with an avian virus, giving us a new pandemic subtype H2N2, but also with a new PB1 polymerase. But the other 5 genes were still from the 1918 virus. 68 switched out the hemagglutinin again, and yet the PP1 again to an H3N2 virus, but still the other 5 virus gene segments are directly descended from 1918. The subsequent seasonal H1N1 viruses that came back in 1977 are entirely genomically derived from the 1918 virus. There are still swine viruses circulating. So in a sense, every single human virus, not the avian flu viruses that people are occasionally exposed to, but every single human adapted virus from 1918 to the present of whatever subtype, H1N1, H2N2, H3N2, are in some ways directly linked to an ancestral 1918 virus. So one way that I've thought about the impact of 1918 is not just that tens of millions of people died in about a year's time in 1918-19 in the pandemic major wave itself, but there are millions and millions of people who have died of flu in the last 100 plus years, all as a result of a single importation of likely a zoonotic event into humans. So, when you think about it in that context, these kind of zoonotic and host adaptive events that lead to pandemics are enormously significant for humans. And so we don't know how long this sort of pandemic era, quote unquote, may last.
It also begs the question of what influenza virus circulated before, right? Because all of that is gone, right? Now we're all derived from H1, right. But maybe there were different strains. Right.
So, yes. So, we and others are keenly interested to know what influenza subtypes circulated in humans prior to 1918. Mm-hmm. Of course, because there are no isolates, the only possible way is to go back and find archival tissue material in which human autopsy tissues would be preserved, wherein people might still have fragments of the RNA of those viruses. We have tried for a decade or more in collaboration with John Oxford and colleagues in the UK, and now more recently with collaboration of my colleagues in Europe and Germany and other countries, to look for more archival influenza genomes, especially those from pre-1918. Unfortunately, so far we have not yet succeeded, but with the newer molecular techniques, I consistently remain hopeful that someday the magic tissue sample will be found, and that will be critically important to take us back, because if we think about it, we have 4 pandemics that we know of out of, say, 15 since 1510, only 4 for which we know anything about the genetic basis of those viruses. So an N equals 4 experiment is, not a huge experiment. Yeah. Even now, we're taking that as a, 100 plus year long experiment. It's still only four examples, each one clearly different, each one formed a different way. All right. So what are the rules for pandemic formation? I have no idea.
Well, I'm glad to hear that you haven't thought it impossible to find pre-1918 genetic material, influenza virus genetic material.
So, I haven't given up hope, but, it's a difficult problem. It's a difficult problem for many ways. In 1918, we were really lucky. We had an incredibly virulent, unusual, one-of-a-kind pandemic with incredible mortality impact, especially in young adults. It was something that made physicians and microbiologists at the time really pay attention. This was something really different. This was weird. It seemed like influenza, but all these young people are dying. So there was an enormous number of autopsies done to try to look at the pathogenesis of the virus. And so there were thousands and thousands and thousands of autopsy studies done, often with tens and maybe even 100 autopsies in these series. So a huge number of autopsies were done in 1918. If you go back and you think about year in and year out pneumonia and influenza, flu seasons, winter pneumonias, people getting bacterial pneumonias, influenza infections, and then subsequent bacterial pneumonias and dying. If you're a physician or a pathologist in 1905, and every year old people die of flu and pneumonia, it's not that exciting. And so I think that there just wasn't that much emphasis on saving that kind of material. Formalin fixation and paraffin embedding, only go back to about 1905. Prior to that, mostly gross specimens were preserved in alcohol, which paradoxically would be better for RNA isolation now than formalin, which does quite a number on RNA, as you know. But those are mostly museum specimens. Here's another problem. Even if these samples exist, the people who own them don't know that they have them. If you call up the chair of a pathology department and say, can I look at your autopsy records and the location in your warehouse of where autopsies from 1889 are, they would go, I don't think we have any. And the answer is probably most people don't, because most of that stuff's been thrown out. It's becoming more and more difficult, but I'm convinced, and this is just my hope driving this, but I'm convinced that there are materials. We just haven't found them yet. And we just, as a worldwide effort, I'm getting a little long in the tooth here. I don't want to be able to be just the only person on earth trying to do this. I'm glad that I have colleagues now here in the US and in other countries that are also engaged in this effort. That's what it's going to take.
You have to inspire young people to want to do this, right? And then they can go on forever. So I don't know if you know, but at the University of Texas, Galveston, I visited in 2019, and they had a huge pathology collection, which was about to be discarded, and they somehow got a grant to preserve it. And they showed me some very old specimens where they kept the entire, say, aortic trunk, everything attached to it, or the lungs, heart, and I mean, it was just huge containers of formalin.
Right.
Might be worth. I mean, I think some of those were very old.
Okay,
and they're in liquid, right? They're suspended in liquid, and some of them are gruesome. I mean, there are all kinds of things, but they decided it was a historical value, and they want medical students to be able to study it. So they're actually preserving it and putting it on proper display and so forth. So maybe you want to contact them. The other thought I had was maybe you should engage Svante Pabo.
Yes.
He works mostly with DNA,
yes,
but
right.
maybe he has
I mean, so certainly there is an overlap with kind of the ancient DNA people, and the forensic DNA people on these things. But, it's not just a question of flu. We have, just as an aside, we have, endemic coronaviruses, 4 of them that have circulated as cold viruses, including a beta coronavirus, OC43, that through some recent kind of speculative molecular mapping has been suggested phylogenetically to have had most recent common ancestor in humans in 1890, where there was a big pandemic in 1889, 1890. So, is it even conceivable that one of those past quote-unquote flu pandemics was a coronavirus pandemic?
Yeah,
sure.
Be, right.
So finding the material is not just useful for flu. I mean, finding 1910, 1920 polio samples, would probably be really interesting.
Absolutely.
Finding old measles, fascinating. These questions are infinite.
And is there any reason to think that there's a small amount of virus in blood, so it might even get into teeth, right?
I think, for measles or, things that definitely are viremic, yes. Flu, not so much, no. Yeah.
Okay. Now, the thing I wanted to ask you before we jump into the rescue is there's this idea that the virus went into pigs in 1918. So tell us about that. There was no influenza in pigs before that, or there was not much pig farming before that?
Well, there was a lot of pig farming before that, starting in the mid-1800s, I guess, at least in the U.S. I'm not sure about around the world. I mean, I know pigs have always been part of, human agriculture for a long time. But certainly there was a big increase in pig farming in the 1800s, at least. Mm-hmm. Cincinnati used to be called Porkopolis in the 1850s. So there was less known about veterinary infectious disease than there was human infectious disease at the time, but there was scholarship.
Yeah.
And most veterinarians at the time said that when pigs started getting sick with classic influenza-like illness like humans, the acute upper respiratory, lower respiratory, high fever, that this was a completely new disease. And there are a number of veterinary scientists who wrote that this was a completely new disease. Okay. And so it's possible that it was, in a sense, a reverse zoonosis, a homoonosis, a human virus then going to pigs. What definitely happened is that the 1918 virus host adapted to pigs in a way that it had its own separate evolution. And one thing that we know about the antigenic drift is driven by pre-existing population immunity. So the people who are exposed to flu have antibodies against older strains, and the virus is selected for mutations that drift. Pigs, unfortunately don't get to live out their natural lives, get turned into bacon, poor things. Mm-hmm. And so there's a lot less population immunity. So what we find is that the 1918 virus in pigs stayed much closer to the ancestral 1918 virus over decades than its human cousins, which drifted significantly. So even Shope in the 1930s said serologically, that when he first isolated influenza A virus from pigs in 1930, said that, serologically, this virus is the 1918 virus adapted to pigs. And I think our genetic analyses would confirm that. It really was. And then that strange lineage of pig-adapted virus, did cause a little scare in 1976 where a handful of people were infected, and there was a thought that it would be the return of 1918 flu, but luckily for us, it was a limited outbreak. There have been just occasional people detected who have contact with pigs who get infected with swine flu. But in 2009, a particular genotype of H1N1 virus from swine that has the hemagglutinin from the 1918 virus through this classic lineage 100 years later, came back into human circulation. And so we've kind of closed the loop that humans gave the flu to pigs, and now pigs gave it back to us, in 2009. So
So despite biosecurity improvements in pig husbandry, the virus still continues to circulate in pig populations.
Sure. And, vaccine control in animals that are susceptible to flu, so that's pigs, horses, domestic poultry, example, are all big issues. Of course, they're regulated very differently than human vaccines. A lot of kind of private vaccines are made, for particular industries. They make their own vaccines. They're not really regulated. I don't really know what's in them. But, we've never achieved really good vaccine control of influenza in humans or in these animals because of the genetic diversity.
And complicating the matter, I guess, is the international pig trade, right? Pigs move around the world and bring their viruses with them, right?
Correct. And, there have been other flu strains that have emerged in pigs. There was a completely independent fully avian H1N1 virus that adapted to pigs in Northern Europe in the late 1970s, totally unrelated to the 1918 and classic swine thing that has been still circulating. Those viruses and classic swine viruses have mixed and matched their genes. They have imported human influenza genes from mixed human swine infections. I think that what I would call a human-animal interface, there must be a lot of what a word for it, like viral chatter going on, right? I mean, the viruses are moving back and forth all the time, but luckily for us, mostly those are kind of dead-end things, but occasionally, and unluckily for us, some random set of circumstances emerge that make it possible for a significant epidemic or epizootic to occur.
Do you think that pig industry is driving flu pandemics?
No, I think despite all of this fear of swine flu and so on, I think pigs have a lot more to fear from humans than the other way around. Not just because we eat them, but because actually humans give flu to pigs, I think, much more frequently than pigs give flu to humans. So it really is more the latter, which is, I think something that most people don't think about.
Yeah.
But, so this kind of biological control of especially, these very high density farm practices that we have now around the world, are clearly vectors for more of this kind of stuff, this human animal viral interface stuff.
It's very interesting because all the ecological disruption obviously promotes spillovers, right? But then all the farming that we do, not just of pigs, but of chickens and turkeys with avian influenza, that's part of it as well. I mean, the humans are really messing things up.
Yes. I think that you could certainly conclude that. So if you think back, so let's say that high-path avian flu viruses, which generally only emerge in domestic poultry like chickens and turkeys, not generally in wild birds. They can spill back over into wild birds, but they generally emerge for whatever reason as there's no good explanation for why that happens, but they emerge in domestic poultry. So if you think back to the old farming style, if you're a farmer on your little farm and you have a flock of chickens and a high-path virus occurs and all 25 of your chickens drop dead in 2 days, you're a mile away from the next person with chickens and it doesn't go anywhere. And so it could have been happening for thousands of years. We don't know. But now when you have, 100,000 chickens in one holding facility and there's movement of those animals back and forth and across countries and across continents, it's very difficult to control. So we certainly must be impacting it. Although if you look at the number of flu pandemics and the timing of them, the world population has gone like that. Our movement between continents is obviously much better. There's much more intensive farming, and yet we don't have any real evidence that pandemic frequency has increased. So while you can imagine that those are all critical factors, nature hasn't actually shown us yet that it's had an impact. I mean, I would agree. I would speculate that it would, but we'll have to see.
All right. I could continue that line for a long time, but let's talk about rescuing the 1918 influenza virus. You talked in the beginning about how you got interested in whether you could rescue material from pathology specimens, and you decided to do a proof of principle with 1918. So tell us how that went.
Right. So Anne Reed and I took on this project to try to find RNA of the 1918 flu in autopsy samples. The FIP had about 100 autopsy records from U.S. soldiers who died of flu, mostly in the fall of 1918. About 78 or so of those had tissue blocks still available. And we started screening them, looking at the H&E slides under the microscope, looking at their clinical records, trying to narrow down those that looked most promising as clinically for having influenza, having died acutely after infection. And for about a year and a half, we couldn't find a positive case, and it was very frustrating.
By pathology, you mean?
No, by PCR. By looking for evidence of viral RNA.
So you would extract RNA.
We would extract RNA and look for. So the amount of RNA that was present was infinitesimal. We tried to make some calculations. There's kind of back of the envelope calculations, but I think the flu RNA in the positive cases is at attomolar levels. We were really down to something close to, single copy PCR. We had to ramp up PCR. I mean, there's no way that you could do it and run an ethidium gel and see it as a band. We had to radio label the crud out of the template so that it was screaming hot so that you could see it. And often if you would make an RNA extraction and then take an aliquot of that and make cDNA, and then do PCR. If you would take 1 microliter of the cDNA and do 3 separate PCR reactions with the same primer set, maybe only one of them would work. I think we were really literally down to sometimes almost kind of single copy in that, microliter, if you think about it. So the other big constraint was that most of the influenza RNA was, tiny oligonucleotides or single bases, which makes it hard to compile the genome. Right. So there's a tiny tail of stuff that went up to about 80 to 100 bases. So what we had to do was to take known influenza sequences, since we didn't have the 1918 sequence, try to make consensus sequence of the most conserved region. So we started with something like the matrix 1 part of the open reading frame, very conserved, and make a little, say, 60-mer amplicon. So we have 20 bases on one side as a primer and 20 bases on the other side of primer that are consensus guesstimate primers. They may be degenerate. And then you might get 20 bases of real 1918 sequence, which we would have to confirm 10 times and sequence in both directions and do multiple times to confirm. Then you could make a primer going in both ways from the real sequence with another guesstimate sequence this way or another guesstimate. So it's like chromosomal walking, but at this microscopic level.
And you have to do it for 8 segments.
So 13,000 bases, nowadays is, it's not a lot of nucleotides, but the way we were doing it, it was a ridiculously labor-intensive task, a true kind of labor of love. We made a vow that we would never have an influenza virus in the lab. We had not been a flu lab, I think was an advantage because, PCR at that level, if there's any contamination, the contamination is going to drive the amplification, so we had no flu. So I had to sit on my hands for a decade thinking about all the things that I wanted to investigate experimentally about pathogenesis and questions of host adaptation with flu while we tried to sequence the bloody genome. So that effort took from 95 to about 2005. Now, using next generation sequencing technology, we can do that in a month. And we are doing that. We're just completing another 3 genomes right now in our lab. But, so when we started, there was no ability to rescue influenza viruses. That happened a few years later between Yoshi and Peter and, George Brownlee and others, obviously developing rescue strategies that would work. And so, it became clear as we were sequencing the flu genome that it would be possible to make chimeric viruses expressing at least one or more segments, and eventually, ultimately, after 2005, to reconstruct the entire virus. And I should mention that we had material from AFIP soldiers. We had subsequently found RNA-positive material from civilians who died in London, contributed by John Oxford and his colleagues. And then a major contributor was unfixed, frozen material from a body of a woman in northern Alaska who died of influenza in November 1918 and who was interred in the permafrost, exhumed by my friend and colleague Johan Hulten, who unfortunately just recently passed away at the age of 96. Johan tried to find the source of the 1918 flu as his PhD thesis project at the University of Iowa in about 1949. Got this idea that if they dug up bodies, they could recover the virus and culture the virus. with mouth pipetting and no lavender flow hoods. And, and luckily, in a sense, for us, that project failed. But his interest in that 1918 flu and knowing that there were bodies buried in the permafrost was that even as a retired pathologist in his mid-70s in 1997, when we published our first sort of birth announcement with some partial sequence of 1918 in 1987 in Science, he was on vacation in Costa Rica and picked up that issue of Science and decided to write me a letter to say, hey, I know where the bodies are buried.
Wow.
And so we did a collaboration, and he was able to get permission to exhume the bodies again in 97, and we found material. So the RNA quality from the frozen material was even worse than the formalin-fixed material, nothing bigger than about 80 bases, but we had a lot more of it. So it became possible to sequence the whole genome from that frozen case. We only have partial sequence from the formalin-fixed cases. Now only with next-generation sequencing have we gone back and done complete genomes of the formalin-fixed cases. So without Johan's help, it would have been very difficult to do the whole genome. But then, of course, in conversation with Peter and Nancy Cox and Terry Tumpy at the CDC and a number of colleagues at different institutions, we put together a big kind of program project grant to finish the sequencing of the 1918 virus and then to do reconstruction at the time. The initial reconstruction and in vitro work would be done by Terry Tumpy in the high containment lab at the CDC. The NIH lab that I'm occupying now was under construction, wasn't ready at the time. So that was not till a year later that I was able to go in and rescue virus.
Wow. And so this is 10-year period that we're talking about here to getting the virus.
To getting the virus, yeah.
And you brought some specimens. You want to?
Sure. But let me lean over. I like to show this now because this is almost 30 years ago now, and this kind of molecular biology is kind of antediluvian, I guess. People, as you know, the young kids in my lab, students and postdocs, you get kits for everything, and you had solution A and solution B and stuff. Where we had to make tris buffers and use a tris pH electrode and all that stuff back then. So molecular biology was just being kind of invented, in the 70s and 80s, and PCR was very new in the 90s. This is I'll just hold this up for you to see. This is a piece of lung tissue that's fixed in formaldehyde and embedded in paraffin wax. This is a very typical look of an influenza autopsy block from a soldier that died in 1918. The amount of tissue is about the size of a small fingernail, maybe a millimeter or two thick. It's extremely limiting.
And if you wanted to look, you would take slices of this.
We would take slices. So the reason that these blocks are is that the way that pathologists back then and still now look at tissues under the microscope are to take, extremely thin slices around 5 microns, about the thickness of a red blood cell, from a tissue block, and then float that piece of tissue and wax onto water and then scoop it up with a glass slide and dry it and then stain it with dyes. And so that's what we would do. We would take these sections, and of course we want to preserve this tissue. It's invaluable, so we don't want to waste it. And so then, as I said, the amount of RNA that we could find was, probably attomolar amounts. So what we had to do was end up running these PCR reactions for these small, say, 100 base or 80 base PCR reactions on a sequencing gel to get good resolution, but also that we heavily labeled the PCR product with P32, alpha P32, so that the product itself was just really screaming hot. And sometimes you'd have to expose these for a week or more, to see the band. And then we would overlay the gel and poke holes or physically cut out the X-ray, and to the gel elute the DNA, clone it, sequence it in both directions, sequence, say, 10 clones or 20 clones in each direction from each PCR product, repeat the whole thing from RNA, 3 or 4 times, and all of that effort maybe to get 20 bases of confirmed sequence. It was an incredible pain, and then running these, ridiculous Sanger sequences, which are beautiful to look at, and they were fun to do, but, a pain, you get a bubble, and you have to redo the damn thing. And again, all this effort, to get maybe 20 bases of sequence of cDNA. So it was, as I said, a real labor, and my hats off to Anne Reed and the others in the team, that put up with doing this project for as long as they did.
Could you scoot in just...
Oh, yeah, I'm sorry.
That's good.
Yeah.
No, fine. So to this block, you would take a slice and extract it. You don't take the whole block and extract it.
Just take a slice, extract it, do a PCR for, say, a very conserved piece of the genome, like the matrix gene, to say it's flu positive or not, and then to go after the experimental pieces that we were looking for. And, the more and more flu sequence that we determined from the 1918 genome, the easier it was to design primers. But, there were still very difficult areas where it was just very difficult to PCR across them. And it took a lot of effort, and then confirming from one case to another. One of the other big problems was that in the mid-90s, the number of flu genomes in GenBank was limited. There were hardly any avian flu sequences, hardly any human sequences.
No GISAID, right?
Right. So, George Brownlee told me that, he got a cell paper in 1980-something, for sequencing the hemagglutinin of PR8, it was a different era. So one of the things that if we wanted to say, where did the 1918 flu come from and how was it related to other flu viruses, was, we need more genomes to look at. So I started advocating to sequence huge numbers of human flu viruses, including archival samples to swine flu, avian flu. So working with colleagues at Tiger and at NIH and Rob Webster and Nancy Cox at CDC and others, we did implement these large-scale sequencing projects that started, around 2000 or so to help us fill in the phylogenetic gaps, and that, in a sense, is kind of it probably would have happened naturally, but I think the 1918 project was, one positive incentive about wanting to sequence more flu viruses. And so, this has all been helpful to try to put this in context. And, so then the last, 15 years or so, has been, our lab and other labs independently looking at questions of origin, pathogenicity, and so on.
So this is basically a single isolate from the fall of 1918, right? My understanding is from reading, there were multiple waves of outbreaks, so similar to the waves of SARS-CoV-2 that we've seen. Is there any reason to think those are antigenic variants, or it was just a matter of susceptibles getting infected like you would expect? Yeah,
we don't really have a good answer. As I said, we have some spring wave sequence. We have some cases from May and some from June and July 1918, the so-called spring or early summer wave.
Right.
And we have hemagglutinin sequence, and that matches the fall wave 1918 sequences. There is one receptor binding domain SNP between 1918 cases, one change with two changes from the avian consensus that gives it really good alpha 2,6 binding and poor 2,3 binding. And the other one that's sort of blended, it's more 2,3, but it has some 2,6 binding. And there is sort of a trend that the earliest cases are more avian-like in their receptor binding and the later cases are more human-like. But it's 10 or 15 cases. It's not statistically evaluable. It's sort of a trend. But we do have early cave cases. Most of our cases are from the fall wave, and we do have sort of a quote-unquote third wave case from London 1919, which is identical in sequence. So what we know so far in these new German sequences that have just come out, full genomes and a couple partial genomes, again, there's very little variation. So I think what we have is that this really was a novel virus for humans. It somehow gained the ability to be transmissible, infectious and transmissible in humans, so that it spread rapidly everywhere, caused the pandemic, and that in the absence of pre-existing immunity, the virus was not under any significant drift pressure. I mean, I guess the early drift pressure would have been to adapt, however long and however that happened. How did an ultimately avian virus get into humans? Was it through an intermediate host? A horse, pig, I don't know. No one knows. But once it got into humans, it seems to have spread at least from Alaska to Germany, whatever that is, 8,000 or 10,000 miles, and have basically hardly any genetic differences, certainly at the amino acid level.
So one of the things you mentioned in the review article with David Morans is that it could be that this is a purely avian virus that in birds randomly evolved or mutated to be able to infect humans and spread. And then it was the right moment where it got into people and it just took off. And as you said, there was no pressure for it to change further. Is that a reasonable idea?
Well, I think it's still a reasonable hypothesis. I think that by any number of measures, the 1918 genome is very avian influenza-like. Its coding sequences are very avian influenza-like. It's phylogenetically avian-like. Its genome content, its GC content is very avian influenza-like. So to get a little bit geeky, the core temperature of birds that are the natural hosts of influenza viruses like ducks tend to be around 41 degrees. And the viruses are probably adapted their RNA-dependent RNA polymerase and their genomes to replicate at a higher temperature and have higher TMs than human viruses, which are upper respiratory viruses, mostly at, say, 33, 34, 35 degrees. And so some kind of adaptation. And so if you look at just the total GC content, nothing about the coding sequences, but the total genomic GC content, avian viruses have a statistically higher GC content than human adapted viruses or mammalian adapted viruses. And 1918 is right up there with the birds. So, people can argue, Eddie Holmes and I and others have talked about this for years and years and years. And the problem is 1918 is the oldest thing to put on the tree. So when you're doing a tree, you can only look at the sequences you have. You can speculate about what happened before, but that's what it is. It's a speculation, right? So we don't know anything about what happened before. It's all under the radar. And I hope
have no avian isolates at all. Obviously.
We have tried to isolate avian flu isolates from the circa 1918 era. After the 1918 genome had been finished and we started doing some flu work out of the Smithsonian, and we have some sequences. I think, unfortunately, some of those sequences probably ended up being contaminants. So I'm not sure that those sequences are all valid, but I have been encouraging people at the Smithsonian and other natural history museums to do that, to go back and look at avian viruses in collections using next generation, non-PCR-based strategies. I think that would be important.
Great. Yeah, that would be. Now, you have virus. You've had virus for many years. And you mentioned earlier this pandemic was particularly virulent. So do we learn anything about that by studying the viruses that you recovered?
I think that we have. I think that what we can agree is that while it was, particularly pathogenic in people, it is particularly pathogenic in experimental animal models like mice, which is unusual for seasonal or human-adapted influenza viruses, which tend to have to be adapted to mice through extensive serial passage before they can become pathogenic.
Like PR8, right?
Yeah, like PR8, which is really now a kind of a mouse virus. The 1918 virus, just without adaptation, is really pathogenic in mice. It's really pathogenic in ferrets. It's really pathogenic in non-human primates. It's just kind of an inherently pathogenic virus.
Okay.
So the logical thought initially would be, since it was such an unusual pandemic, there must be, some key set of mutations that made it so pathogenic. So if we could align it with other sequences, we could go, aha, eureka, here's the mutation that relates to it. So through extensive work, what we can say is that it's a really good flu virus. It replicates really well. It has a really efficient polymerase. It also expresses a very avian-like H1 subtype hemagglutinin with hardly any non-synonymous changes from a typical avian consensus H1 you would find in a duck pond, half a mile away from here. So, except for these receptor binding changes. But so, in my lab, we did an experiment about a dozen years ago or so with Chi Li. And what she did was she took the 1918 genome and she made a set of rescue viruses in which we replaced one of the 8 gene segments at a time with the sort of a typical low-path avian influenza virus from wild ducks. So no mammalian adaptation, with the idea that, that would certainly tell us something about host adaptation and possibly virulence. What you find is that 7 of those 8 gene segments, in which 1918 has 1 avian gene segment, do not attenuate the virus at all. And only 1 did, and that was PB2. And we know that the PB2 at its C-terminal end has some changes that are important for getting the RNA polymerase into the nucleus, since flu virus replicates in the nucleus unusually. And that changes a glutamic acid to lysine change at amino acid 627. And human viruses, including 1918, have that, and avian viruses tend not to have that. And if you make that one nucleotide change in the avian PB2 segment and put it in with the rest of 1918, it is 1918. So then we thought, well, that's just crazy, because we knew that the polymerase was an important factor, but we also had very good evidence that the hemagglutinin was a big virulence factor. Because if you took just the 1918 hemagglutinin and put it on the backbone of a seasonal virus, which causes no disease in mice, it kills mice. It kills ferrets. So the hemagglutinin was a major factor. We started by making these chimeras with avian H1s, and so we would take the HA1 domain and the HA2 domain, or, the untranslated regions or the transmembrane domains, and, make these chimeras to say, well, maybe we could narrow down where the changes are. And every chimera was just as pathogenic. So we tried a whole bunch of avian H1 viruses. As long as they're on a mammalian adapted backbone, they're as pathogenic as 1918. And one of those features is that they induce an extremely strong neutrophilic infiltrate. Very early on in the lung. It's really unusual for RNA viruses to induce a strong neutrophilic infiltrate. Mostly it's a, monocytic, lymphocytic infiltrate. And so 1918 characteristic is this huge number of neutrophils in the lung, seen in 1918 autopsies. That's what you see in mouse lungs, we see in non-human primate lungs and ferret lungs. But what we now conclude, and what I think, is that its virulence is an accident. The accident was that an avian virus that is not pathogenic in its native host in birds is accidentally pathogenic in a pro-inflammatory way in mammals and in humans. And what we now know through extensive studies where we've looked at H1 through 16, all the different subtypes of hemagglutinin in birds, and looking at this, is that H1s share the feature with 1918, but also H6, H7, and H10. So when H7 and 9 started causing human outbreaks, I was very concerned that it has the kind of characteristics of high mortality that the 1918 H1N1 had. So I now think that if you think teleologically about adaptation, if an avian virus is, quote unquote, wanting to, and I'm sorry for being anthropomorphic, but wanting to adapt to a mammal, being able to replicate in those cells, infect those cells efficiently, to make progeny virus and then transmit them from one host to another would all be areas where there would be strong selection pressures. But if a flu virus, which has its peak replication at a couple of days, maybe shed the virus for 5 days, and then is no longer present, if you then subsequently get a secondary bacterial pneumonia and then subsequently die at day 12 or day 14, again, speaking anthropomorphically, what does the influenza virus care if you've died if it's on to another person? Not to be crude, but it seems that to me that there would be strong selection pressure for host adaptation for physical things, infectivity and transmissibility, but very little selection pressure for pathogenicity or inherent virulence, since it's a kind of a blitzkrieg virus. And I think that we see similar things with SARS-CoV-2 that in the autopsy studies we've done and others, you see the same kind of very strong neutrophilic response, very strong pro-inflammatory response that I think is a huge contributing factor for pathogenicity. Another experiment that our lab has done, John Cash led, is that if you treat mice with a free radical oxygen inhibitor as a way of kind of muting neutrophil response, it has no antiviral properties. It doesn't affect viral replication at all. You can infect mice with a lethal 1918 infection. Those that are untreated all die. Those that are treated survive. Their viral titers at every point in the infection are identical between the two groups. And so it's not viral replication alone. It's clearly the host response that has a major contributor to pathogenicity. And so I think that's for me, the big lesson of 1918. And the other big lesson of that is that 1918 is not unique in that there are other avian viruses out there silently in the world that could share those features of other subtypes, and that if those were to adapt to humans in the future, we could see a similar outbreak.
When you mentioned the list of avian hemagglutinins, you didn't have H5 in there. Does that do something different?
H5, low path H5 is not pathogenic in this way. High path H5 and high path H7 have an enhanced cleavage site, a furin cleavage site, like we people have been talking about with SARS-CoV-2 that other coronaviruses also share. That allows the virus to replicate systemically throughout the body. So in the case of H5, the pathogenicity is very pathogenic in avian species particularly. I mean, when people say high-path avian influenza, they don't mean it's high-path for humans. They mean
birds.
Chickens and other species of birds just drop dead from this virus. There have been, of course, a small number of zoonotic infections. Luckily, those cases have been relatively rare, and those viruses have never human adapted. But in the absence of that furin cleavage site and the ability to replicate, say, in the brain or systemically, and then the H5 hemagglutinin itself doesn't seem to share those properties. Now, we stopped working on the molecular basis of that HA pathogenicity with the gain-of-function ban a dozen years ago or so. And then we started doing other things, like we're working on universal influenza vaccines and started lots of clinical studies with influenza. And we're just now getting back to flu pathogenesis again, and then COVID happened and our lab became a full-time COVID lab for a few years. So now we're just getting back into this question of HA pathogenicity, but this is a very important question, and the basic science of this is critically important, I think.
So is it the case that a virus in a bird randomly changes such that if it entered humans, it would trigger excessive inflammatory reaction for reasons we don't know? Or is it the case that to do well in birds, those changes then provoke this abnormal response in humans? I mean, those are two possibilities, but I don't think you could distinguish.
I don't think you could distinguish them either. We have so few examples. We know of no H10 mammalian adapted things, but there have been some avian H10 outbreaks of minks in Scandinavia, and they have been incredibly virulent. They had really high mortality. We know that H7, human infections of avian H7 and 9 in 2013 through 16 or so, were pretty pathogenic. So there are some hints that those subtypes that we find experimentally in mice and in human cells in culture, are inherently more pathogenic, but they don't seem to be pathogenic in birds. But very little has still been studied of flu, the natural kind of biology of flu in their natural hosts, wild birds and ducks. It's mostly GI virus, not so much a respiratory virus. It seems to cause no apparent disease. If it gives birds the runs, how would you know? Because,
they always seem to have the runs.
And so, it seems to be a very well-adapted virus for them.
Right.
Multiple infection with multiple subtypes seem to occur simultaneously. It's not really clear how and if they develop good long-term humoral or cellular immunity against viruses. There are so many mysteries of how flu evolves. I think it's just clearly very different in different species. So the evolution of flu viruses in birds, I think, is not as complete genomes. I think that they're so adapted, and the segmented genomes, I think they're so adapted to their native hosts that if reassortment occurs, if a double infection occurs, as long as you have all 8 segments packaged to make a virus, it's a virus. So it's like, imagine building a virus with 8 different Lego blocks with 8 different colors. As long as you have a blue one, a red one, and a green one, and a yellow one, and a brown one, they just work. They all work. Now, that's not to say that every reassortment combination is just as good, but, they all seem to work. So there's massive reassortment. So when you look at the phylogenetic tree of avian influenza virus genomes, and you say, oh, this is an H5N1 virus, the H5 is gone there, and the N1 goes there, and the PA goes there, and, they don't stay together. So you can trace individual genes, but they're always in different clades. So I think of it as a kind of a gene pool. Flu is kind of an RNA segment pool in birds, and that on the rare events when it leaves that pool and goes to a non-native host, like a pig or a human or a horse, you now have a single virus with a single 8-segment genotype that has to survive on its own in this wilderness of this new animal. And it has to invent new ways to evolve, which are to undergo antigenic drift to be able to reinfect that species again and again and again. If a H1 virus infects a bird, maybe it does develop at least transient anti-H1 immunity, but then the virus doesn't need to undergo H1 drift. It just puts in an H10 and then an H13 and then an H4 and, and then the duck dies when it's, age 5, and so the virus has so bird viruses are incredibly diverse between subtypes. The average inter-subtype identity, say between hemagglutinin subtypes at the nucleotide level is only around 35%. I mean, unbelievably diverse. And yet there's never anything in between. you can say that H2 and H5 are genetically related. They must have come from a common ancestor. But nowhere have we ever found a half H2, half H5 virus. So Eddie Holmes and I have speculated that there was some kind of fitness threshold, and that as these viruses diversify their subtypes, those ancestors get selected against, and now we're only left with the ancestors. But that is a very dynamic process, and that while we think that there are 16 hemagglutinin subtypes in birds now, maybe 200 years ago, it would have been different, and 200 years from now, it's going to be different. And that we have this mindset that just because we've sequenced these things and we can look at them on a tree, that's the way Mother Nature made them, where it's a really dynamic thing. So I think that they're constantly evolving, but in a completely different way than flu viruses evolve in mammals.
So that leads to my question of, do we understand why in humans we only see H1, H2, and H3, and not all these other hemagglutinins?
No. Matter of fact, I think the simple answer to every question you could ask me about flu is that we don't understand anything. After 30 years of thinking about it intensively, I now think that I know enough about flu data that I understand absolutely nothing. And I'm completely comfortable with that right now. That's my kind of new level where I don't understand anything. Flu knows what it's doing, so to speak, but we don't. And it's really humbling. But is that because there is some kind of magical property? I mean, and by magical, I mean some physiological property that we don't understand, that of the 16 subtypes available to us, only H1 and 2 and 3 can adapt to humans. Okay, so let's look at pigs. Pigs have mostly H1 and H3 viruses, but mostly they're human-derived. There are some avian H1s. Horses used to have only H7s, and that has gone extinct, and now they have H3 viruses and a couple lineages of H3. So if you take humans, swine, and horses, H1 and H3 seem to be common. So does that say that there is a biological constraint? That H2 only circulated for 11 years. So maybe you could argue it was kind of a loser. It didn't, it didn't have what it took to make it. I don't have any firm ideas. I'm back to the N=4 experiment. We have no idea what subtype caused the 1510 outbreak or, the 1788 pandemic. We don't know. We don't know what the future pandemic will be. So I always bet on the virus. They're very clever, and humbling. And so I don't know that we can just have this rule. There was this whole idea that there was this a pattern of circulation. It went H1, H2, H3, 1, H2, and 3. And it was because it was an N=3 experiment, and so they said, well, that's what happened. So we had H1 and we had H2 and then we had H3. And they're called that just because we only discovered the different hemagglutinins after the pandemics occurred. But I don't think there's any data for that. The old serology data were after the 1957 pandemic and they figured out that H2 was different than H1. They didn't call it H2 right at the beginning. They went back and looked at serum collected before the pandemic, and they said, people alive before 1918 have reactive antibodies in their serum to this pandemic virus that no one's seen. So before 1918, must have been an H2. And then people did that when the 1968 pandemic occurred. And so in the late 60s and early 70s, they looked at old sera collections, and they said, look, they have reactive antibodies to the H3. So what happened in 1918 must have been an H3. So half the literature says the 1889 pandemic was H3, and half of it says H2, and it's usually N question mark.
Okay.
Serology, even at its best, is kind of a mystical science, and cross-reactivity and so on. So the only way to know is to go back and get sequence information of the virus. So, but what that will say about future pandemics, I don't think we can predict.
So it's interesting that you say this about the N conundrum, because when I was trained in influenza by Peter and Ed Kilborn in the department, you have H1 and then it goes away, then you have H2 and it goes away, and then you have H3. But now we have H1 and 2 and 3 co-circulating in humans, which blew me away when I first heard it, because that's not the way I was taught. So it means there are no rules, really.
There are no rules, right. Every pandemic's completely different, the way they form, the way they emerge. We think that pandemics, back to our very first question, what is a pandemic? A pandemic, you think of it as a significantly impactful virus, that is a virulent virus. But even Shope, in a good lecture in the mid-50s, said, a pandemic is just a novel virus. It has nothing to do about its virulence. So, the 2009 virus wasn't very virulent. If the 2009 virus pandemic had occurred in 1730, would anyone have distinguished it from seasonal flu? I doubt it. So how many pandemics have we missed because they're not much different in terms of their public health impact? Mm-hmm. But I think that the problem is that we're so limited in our information, and also there's no periodicity. So if you look at what we think are influenza pandemics going back 500 years, the span goes from 11 years between 1957 and 68 and 175 years back in the 16 and 1700s where there's no evidence of a pandemic. So, if you average it out, it comes out to about 35 every 35 years, but it's incredibly variable. Pandemics kind of happen when they happen. And why did H1 go away in 1957? Why did H2 go away in 1968? Why did H1 survive upon its re-emergence in humans in 1977 and has kind of competed for the niche with H3? Why is that? I don't know. And what is the impact of COVID on flu circulation? Right? So there's been limited flu circulation for mitigation measures we've done for COVID. Flu is just sort of beginning to kind of come back, and maybe that's another measure that COVID pandemic might be ending if we start seeing more endemic virus circulation again. But if very few viruses were circulating, has that limited the gene pool? Of the two flu B strains that are circulating, one of them has basically disappeared. H1 viruses are really rare. Would they go extinct because of COVID? I don't know. Will they just come roaring back? I don't know. The 1968 pandemic, when emerged, a clearly novel virus with a novel HA subtype, was not very pathogenic. That virus has now circulated for almost 60 years in humans, year after year. And in a sense, it's becoming more pathogenic. The number of people who die of H3 infection as seasonal flu now, 55 years later, is much greater than what happened in 1968. Why is that? But H1 viruses seem to have attenuated. None of it makes sense. I mean, there are I firmly believe there are explicable biological reasons for all of this.
We don't know them.
But they're super complicated, right? So, your flu virus exposure history, your immune history as you go through life, people have talked about this idea of imprinting. It's a big idea in the field now, right? That what you were exposed to at first might relate to your ability to respond immunologically to other things. Right. There's some data for that, some data against that. It's still controversial, but, all of these things play a factor. And so if you look at time, not only do influenza virus change over time, the human population changes over time. The age demographic changes, our exposure history to flu viruses change, and so it's an ever-moving target.
I was disturbed by reports that viruses become more virulent because it seems counterintuitive. But then I did a TWiV with Catherine Hanley a while ago, who said, well, if reproduction levels increase to allow better transmission, that could, as a side effect, increase pathogenicity. Right?
Absolutely, yes.
It may not be what is initially selected for, but that could be. So maybe that is what's happening with H3N2.
Right.
Who knows? Right.
That's certainly possible.
I'm worried about H2N2. I talked to Ron Fouché a couple of years ago, and he says it's in birds. You can find it in birds. So now the population immunity is declining to that particular virus, so it could re-enter the population, right?
Right. Yes, I think H2 is something to always worry about. We know that it worked. We know it adapted to humans. The one sort of saving grace is that the neuraminidase subtype, the N2, has been preserved in the 1968 H3N2. And obviously, it's massively drifted, but, H2 would worry me. One thing that, in addition to a natural origin, but something to say, is that there are H2N2 isolates from the human pandemic and seasonal strains between 57 and 68 in freezers all over the place. Sure. It has not been a controlled virus, not like smallpox or something. Only recently has the U.S. CDC recommended, that it be worked with a BSL-3 and under, careful conditions. But, it's certainly possible that in an accidental release of a virus like that could could circulate again.
Sure. Yes, Ron says his freezer is full of H2 isolates.
Right. Luckily, all my H2 isolates are in the BSL-3 freezer. So
I'm amazed how little we know, given all the work that people do on this virus.
It's really depressing. I mean, you talked about staying hopeful, but, you think influenza has been intensively studied for, almost 100 years now. And, all your basic questions, we still don't know the answer. I can send you, there's a monograph written by a German physician in 1820 that I found. And he's writing a book about influenza in Europe. And on the title page, he lists a series of questions that he wants to address in the book. And his questions are, where does influenza come from? Why does it circulate when it does? How does it cause disease? And what can we do to prevent it? And those are damn good questions. And those are the exact same questions that are just as relevant today, 200 years later. And we have lots of blah, 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 genome, blah, 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 immunology. And when it comes down to it, we don't know anything more than he does. It's sad.
Well, it's even worse for the coronaviruses, I think.
Right? Absolutely. Understudied. Massively understudied. Well, at least up until the last 2 years ago. Where now half of PubMed is SARS-CoV-2. But
yeah, it's amazing, isn't it?
It is.
But it hasn't outstripped influenza virus genomes, right?
No. Well, it must have. I mean, there are millions and millions genomes now.
Remarkable.
Yeah, it is remarkable. But still, all the basic science questions of, coronavirus adaptation and genome complexity and, pathogenicity obviously are way behind where we are with flu.
It drives me crazy when a new variant of SARS-CoV-2 emerges and people immediately say it's more or less pathogenic when you can't learn that so quickly. And we still don't understand it for influenza viruses. We have animal models, we have human observations. It's not that easy. You can't do it on a hospital observational study, right?
Correct. Plus, at the same time, people have exposures, prior infections, at least exposures, vaccinations, and immunity. So, how do you separate individual and population level immunity and emerging antigenic drift? And, it's impossible, right? And our animal models are limited. Obviously you have to use kind of ACE2, humanized ACE2 positive mice, other animals, but none of them are mimicking severe disease in human COVID, to my view.
No, they don't.
And so I've talked in our lab, we've been seriously, looking at COVID and looking at COVID response in terms of vaccine development and pathogenicity studies through autopsies. And we would very much like to approach some of the basic pathogenesis questions we have with flu, but it's extremely challenging. And, in mice, it's mostly an encephalitic disease. It's not really a pneumonia, where it's not in humans. And so,
How is a ferret for COVID? Is that any
terrible?
Better? It's terrible.
Yeah. Ferrets just aren't susceptible. Ferrets are susceptible to everything, are just not really susceptible to COVID. It's so weird. Their ACE2 is just really different.
I remember when Omicron first emerged, there was a study out of a South African hospital. Oh, look, it's mild. Oh, but we forgot to say the population they're all young. Mm-hmm. The old people have died. They're mostly immune. Mm-hmm. And so the press just picks up mild, and Omicron suddenly became mild, whereas it's not, and it still kills people, which is really a reflection of the difficulty of communicating in an emergency situation, where people want answers and science doesn't work that quickly. Right.
I can say just on a personal level, as a patient rather than as a physician, I've had COVID twice, despite the vaccine regimens. Mm-hmm. Once before the vaccines were available, early March 2020, and it was incredibly mild, just a couple of days, sore throat and nothing. But I had a huge spike in antibodies that I tested in the lab. And now I just recently, post-boosting, have had the BA.5, and I was sick for days with 103 fever and wheezed for 3 weeks. So that the mild Omicron BA.5 infection was, in me, N equals 1 experiment, very severe. Why is that? I don't know. But it is. So yeah, it's very difficult to make. That's not difficult. It's easy to make assumptions. It's bad from a scientific standpoint to make these assumptions, without data. So we should always be very judicious about, how we approach these things. We're all going to kind of live this in real time, right? I mean, what happens to COVID? I don't know. What happens after BA.5? I don't know. Does it become an endemic coronavirus? Does it become more like a cold virus? Does it stay like kind of a mid-level pathogenic virus like seasonal flu? I don't know. Does it go extinct? I don't know, right? Those are the answers. That's the answer to give. I don't know.
The press finds people who give answers. That's the problem.
Yes.
Right?
Yes.
If they came to you and you said, no, I don't know, they wouldn't publish your interview. Right?
Well, that's what happens all the time, right? I mean, if you try to not speak in geek language, but you try to say, look, we really don't understand these things. It's really complicated for this and this and this and this reason. It's like, there's no headline. It's boring.
No,
right?
No, they will find someone who says, this season's going to be horrible. We're going to have new variants that are going to evade vaccines, where there's no evidence for any of that, but that's what will get headlines, and then people get scared.
Yeah.
And so I think the role of scientists is to try and mitigate that and say, look, we have no idea. We're playing it day to day, essentially.
Yeah,
right?
A completely novel virus. Like you said, we had no information about it, and now 2 years later, we've learned a lot, but we still have so much more to learn. Yeah, it's difficult. I think communicating science is becoming more difficult now. I don't know. I'm getting into fields that are outside of my bailiwick, but with a kind of a 24-hour instant news cycle, with social media and stuff where everything's instantaneous, everybody is an expert.
That's the problem.
Or the experts are discounted because they're biased.
Yeah.
Oh,
absolutely.
And it's this kind of crazy world.
Totally
and so
right.
I don't know how you communicate science effectively. I mean, obviously your podcast and others like it are really valuable. And there are really good science reporters around. And I know that our institute tries very hard, to communicate science effectively, but it's difficult. And I think the environment's just getting harder.
You've hit all the right points. Expertise is undervalued. In fact, have you read The Death of Expertise, the book?
I have not.
Tom Nichols, highly recommended. He tries to understand the sources of this death of expertise. Very good. And only one is, the internet is part of it, but it's not all. And that people think they can become an expert by doing a few Google searches. And so who cares that you worked on viruses for 40 years?
Right.
But it is a real problem. I mean, what we do is simply give you the science, and we attract a certain number of people, but by no means millions of people, right? Because they don't like, we're too long and we're too complicated, but we attract the people who value the good science. And I don't know what the answer is going forward. I'm just going to keep doing it and try different things. And I get scientists to talk about their work, and I think that there's a lot of value in that. So
my wife gave me a coffee mug recently that says, "My medical degree is worth more than your Google search."
That's good. Yeah. I like that. It is.
Yeah, it is. It's more expensive too.
But there are people who will
argue with that. Yeah.
I did a podcast with Lex Friedman a couple of years ago, who was a very well-known podcaster out in Austin, Texas, and he does all kinds of areas, and he has a lot of followers. And he said to me beforehand, don't tell us you're an expert, please, because we don't really care. And I was like, don't you care I want experts on my program, right? I don't understand this at all. But that I might be too old. I don't know.
No, I don't know.
I want to just finish up with a couple of influenza virus questions. And so three. First, and these come from your review article. So one of the questions is, how do we study pandemic potential, right? As you have said, there are a lot of influenza viruses out there. Should we focus on H1, H2, and N3 only and see where they are? So give us some thoughts about how to do that, because we should be thinking about what might be the next virus, even though you might say we have no idea.
Well, the answer is we have no idea, but we should be thinking about the next virus, and we should do our best. So, I think increased surveillance, especially at the animal-human interface, is crucial. And I think that, it's not unreasonable to look for H1, 2, and 3 viruses as future pandemic viruses, since we know they've happened. But the thing that would go against that is that H1 viruses have been in humans for more than a century, H3 viruses for now almost 60 years, 55 years. There's a lot of population immunity. So H2 of those 3, from that standpoint, from a population susceptibility standpoint, is high, but there's zero population immunity to H11 and H4 and H8, and right? So I don't know. I thought ignorantly, as a young person, that if we sequence the 1918 virus and we find the mutations that have it differ from, its ancestral avian-like viruses, we solve the mystery not only of host adaptation and virulence. And then you find that we have identified a key number of amino acids that are responsible for host adaptation and pathogenicity in the 1918 virus, but they are unique to the 1918 virus. The 1957 and 1968 viruses have different changes. The 2009 virus, different changes. Matter of fact, one of the key changes in 1918 associated with host adaptation, if you put that experimentally into the 2009 virus, attenuates it. Where in the case of 1918, it causes it to be unable to replicate in mammalian cells. So, what we really need for all of this is a much better understanding of a genotype to phenotype correlation. It's very easy to align sequences, genome sequences or their translations and say, oh, look, here's a non-synonymous mutation here in PA at residue 153. But what is the structure of PA and how does that relate to the heterotrimer, to the polymerase heterotrimer with PB1 and PB2, and how does it function? And flu viruses have very particular needs. They need to be able to replicate in cells. They need to take over your machinery. They need to import themselves in their segments and the polymerase complexes, proteins, into the nucleus to allow replication to occur. They need to package themselves and all those things. So there are a certain number of constraints that are going to happen. And if you're going to do that in a new animal, you're going to have to adapt. But maybe this change here in one situation solved that problem structurally for them. And another change here solves the problem structurally in the exact same way because of the way it's folded or it's three-dimensional, it's multi-dimensional interaction with other viral proteins or host proteins. But we're so stupid, we can look at this mutation and say, well, they're different. But, they're different because they're different mutations at different sites, but maybe they're doing the same thing. What we need is a much better understanding at the structural level. I mean, for heaven's sakes, there are only about a dozen proteins encoded by influenza A virus, and we still don't know what they all do. I mean, that's crazy. And so, then you think of, a big genome like SARS-CoV-2, much bigger with many more open reading frames, and you go, right? So
people just look at Spike.
People just look at Spike, right? But, this is the thing. It's super complicated. And so my naive hope that we would have a checklist. These are the things to look for when we want to screen for the next pandemic virus. I now feel that all of that's just no good. And that we're never, at the moment, I do not think that we have enough information to predict the next pandemic, but we can react to the next pandemic. And the question is, how quickly can we react? And that's important. So if we have better public health systems, if we could make quote unquote universal influenza vaccines. I know it's a topic we haven't discussed, but vaccines that would be effective that are not specifically matched to a particular virus isolate. Right. Strains that would give us broader protection, even if it's not great protection, some protection. If an unexpected virus emerged from birds tomorrow and started circulating in humans, it would, at best, it would take months and months and months to make a vaccine. So it's always sort of too late. So if you had a vaccine that could offer at least some protection, some mitigation to the spread, the pandemic, that would be good. Better effective antivirals would be really useful, other therapeutic interventions. So I think that we should prepare for the next pandemic because I feel a 100% certainty that we will have future flu pandemics, but I have no idea when they will occur, where they will occur, what subtype they will be, and how pathogenic they will be. But you can do what we can do, right. Starting from Shope 100 years ago to us and to the next generations to follow. I mean, that's what we have to do. We have to continue to study these viruses that are, have such huge public health impact on people.
Do you think it's reasonable to think we might have a universal or some kind of broader flu vaccine at some point, or is that going to be impossible?
Well, I think it's a very difficult task. I think that there are lots of problems. I mean, flu is this master at antigenic drift and mutation, right? Positive selection. It's never standing still. We have to reformulate the vaccine every year to keep up with that drift. And then with new subtypes and pandemics, of course, it's even much bigger problem. But if you look at other respiratory viruses like RSV, sure, there's genetic variability, but not like flu. They don't have the ability to, their surface proteins aren't plastic like flu, or measles is another example, And so in the one case, measles, you get really, really good immunity through a natural infection or through vaccination. Flu, you get not so much immunity through vaccination. But even through natural infection, people can be reinfected. Now, they're being reinfected with different strains. But in a small clinical study that my group did with Matt Memley here at NIH, we found that in a challenge study, a handful of people, less than 10, so it was just a very small study, that they could be reinfected with the genetically identical influenza virus 1 year later. And that the majority of them were able to be reinfected. So these are young, healthy people who were infected with the virus, mounted a B and T cell response, cleared their virus, had no problems, and then were reinfected a year later, and they were still able to be infected.
But did they get sick?
They didn't really get very ill either time because they're young, healthy people with an H1N1 virus. So it's hard to say. I mean, obviously, the study's not designed to look at pathogenic viruses, obviously, for ethical reasons in people. Sure. But the point is that you don't have sterilizing immunity after 1 year when you're a young, healthy person against the genetically identical virus. I think that viruses that only replicate in the mucosal surface, like flu, mostly like COVID, like RSV. I mean, how many times do you get infected with RSV from a baby to when you get an RSV pneumonia as an 85-year-old and die?
Many
often,
times,
right?
Yeah.
And I think that we're missing something. We're missing something about memory responses for the respiratory mucosal system. The concentration has always been on systemic immunity, right? How is a vaccine evaluated? Your serum antibody titers against HA. The flu isn't in your serum. It's not circulating. It's right here. What are the mucosal correlates of protection? But I think another concern that I have is the rapidity of the viral replication. So let's say that you do have good immune responses. You have a memory T cell response, you have memory B cell responses. You have to dredge those up. They have to migrate to the lung. It takes time. You have to gear up. You have to copy. By the time it's done, the flu says, see you, bye. So I think that we can certainly make progress. And I certainly think that we can make better super seasonal vaccines. But a true quote unquote universal vaccine that would, one shot for your lifetime, every flu, avian flu, horse flu, pig flu, whatever, I doubt it. But I think a reasonable goal would be a vaccine that offered protection against serious disease and death would be really valuable. Not necessarily a neutralizing vaccine. I mean, that would be great, but I think it's too high a bar. So if you had a vaccine that if a new pandemic or a really weird strain emerged and it gave you some cross-protective immunity, say a T cell epitope that's not as, variable as the hemagglutinin protein, that it prevents you from having to be hospitalized, prevents you from having to be ventilated, that's a big win. So that's what I think. I think better vaccines can be made, but I think we have to focus on mucosal immunity. I think we have to understand immune correlates, both systemic and mucosal, and the interplay between the systems. I mean, the immune systems, I'm not an immunologist, but as I'm learning, the immune systems are really, while they have interconnections, they're really kind of separately functioning and separately developing. So, how do we cross and talk between them? So maybe the emphasis on just, IM vaccination isn't the right approach. Maybe we should be thinking about other ways, intranasal or mucosal vaccinations and, or combination vaccines.
IM is what we know how to do. FluMist has not been a great saver.
It hasn't. Right? It's probably over attenuated. I mean, it sounds great. Obviously other live attenuated vaccines have been great, but the FluMist sort of hasn't, it's a complicated thing. It's sort of like the dengue, you have 4 viruses and you have to balance that. And, it's complicated. It just unfortunately hasn't been as good as it should have been.
I think that the idea that a mucosal vaccine will necessarily be better at blocking infection isn't all that supported by the immune system, right? Antibodies and T cells go down after a few months.
Correct.
It's not physiologically good to keep them high, right? So as you said, if a virus is replicating quickly, by the time the memory response kicks in, the virus is gone. And so I don't know if you've heard recently, people have been saying if we don't make a nasal COVID vaccine, it's a biosecurity risk because other countries have got it. But they don't prevent infection. They prevent disease, maybe.
Maybe.
So, but I think the idea of somehow inducing a T cell response that protects against severe disease is great. And I think that's why the ancestral COVID vaccines still protect you against severe disease.
I agree. So I think that, if natural infection, say, as against flu, as we said with RSV or other things, doesn't prevent you from being reinfected, it's hard to imagine how we're going to do a vaccine that's better than a natural infection.
Exactly.
But we can learn from natural infection. And here's what we can learn is that there's such an emphasis for the last few decades in infectious disease research and vaccinology on monoclonal antibodies and finding magical epitopes, five amino acid things that are the key that's going to neutralize every flu virus, right? It's a nice dream, but that's not how your immune system responds to a flu infection. You make antibodies against every epitope presented. You make T cells against every T cell epitope that's presented. Systemic immunity, mucosal immunity, you get memory cells. Maybe you get memory resident T cells. Maybe you get memory resident B cells. You certainly get resident memory B cells and T cells systemically. But as you said, it's not good enough because by the time you mount that up, the virus is gone. If as you said, if you maintained high serum antibody titers to every antigen presented, I mean, your serum would be sludge, right? You'd have multiple myeloma, right? I mean,
yes,
obviously
exactly.
that's not physiologically good.
That's a great point, yeah.
So, they have to be downregulated. So, maybe something like a compromise, maybe some kind of vaccine strategy that might involve a mucosal administration, but maybe it happens every season. So in a dream world, something David Morens and I have discussed is, what if you had some kind of reasonably good vaccine that could be delivered mucosally, say an intranasal spray of whatever formulation, but that it gives you some kind of breadth of protection that wouldn't be strain specific. Even if it works really well, maybe that's not very durable. So what if, you go to the pharmacy and you get off the shelf, this little spray bottle, and at the beginning of flu season, you squirt it up, and then in January again, you squirt it up again, and you boost your immunity a little bit, and that maybe you're not achieving a universal vaccine, the one magic shot that you get when you're 5 and, like measles, but maybe there are ways that we could mitigate infection. But again, to stop infection, to stop transmission of these viruses is just really super challenging. And it's a great goal. We should all be working on it. I'm working on it. Everyone's working on it. But I think we should be realistic. It's hard.
Some people say, if you don't try, you're never going to learn, right? So even if we say the immune system works this way, there's no way around it. Okay, but maybe if someone has some good ideas.
Right. And we've been making flu vaccines the same way for 60 years. And it works. And if the strain is matched to that year that, a year later after the vaccine is made and you get decent protection for that year. And if the vaccine is a mismatch, you get lousy protection.
Right.
But there's no reason why we should continue to make flu vaccines this way. I mean, we're in a new era now. We're in a, molecular era. We can think of new platforms. We can think of new delivery devices. We can think of new strategies of immunization. We can think of new correlates of protection through studies of, human immune systems. Just because we have been comfortable doing it for 60 years, to me, and I'm speaking as a person, not as a representative of the United States government, why should we be comfortable with a vaccine strategy that hasn't been very effective, not effective enough? So I agree with the push for new generation vaccines. And it's great that there are 200 different ideas out there. That's the way science should work, right? And people who disagree with each other and who think their other competitors' model sucks and, all that stuff. That's good. That's right. That's good. Because, together, collectively, we'll begin to learn something, right? That's the way science works. So that's what we need to do. We just need, a kind of a kick in the rear to do this. And maybe, the push for universal flu before COVID and now with COVID, maybe that really gives us the permanent kick in the rear to do this.
And also COVID has exposed some vaccine approaches that were kind of not really looked at, and now they've been extensively. So I understand a flu mRNA vaccine is being tested, right?
Right, as it should be.
That's fine, but maybe we could go beyond the HA, right?
Exactly. So, I mean, that's why the approach that I'm taking, and we've recently published on, is an approach in which you get a whole virus, an incredibly simple idea, a whole virus inactivated vaccine, but with a mixture of 4 different avian influenza viruses with 4 different HA and 4 different NA subtypes. But it's BPL inactivated so that you don't get much antigenic change like formalin-fixed vaccines. It's mostly the RNA that's mutated. So it's really a kind of a non-replicative virus, but it's presented. It still has HA activity, has neuraminidase activity. It's taken up by cells. what we find is that we get incredible cross-reactive immunity with a very complex immune system. We get systemic antibodies, we get T cells, we get mucosal IgA, we get B-cell, T-cell responses, but we get incredible protection against influenza viruses expressing subtypes not in the vaccine at all. So that's nice, but, as Peter Palese says, it's in mice, it's in ferrets, it's not humans. So we're in the middle of doing a phase 1 right now. So far, that's ongoing. It's still blinded, but obviously from a safety standpoint, everything's going well. The safety profile in animals is great. The real question will be, do we have efficacy in humans? And the only way to evaluate that is in phase 2 type efficacy studies, and that depends on the funding, and it's all very expensive. But, these are the things. So that idea is one idea. Nanoparticles are one idea. mRNA is one idea. Stock-based thing. We should do everything, because one of them is probably going to be better than what we have. Right, but it all takes money. Yeah.
It takes money. We need more scientists, too.
We need more scientists, yeah.
I think we don't have enough.
And, doing science is harder and harder, right? I mean, the age of getting your first R1 keeps going up. And, we have a lot of leaders in science who, generationally don't retire, right? And so there's not much upward mobility right now. There's some very discouraging aspects about science. Think of the training you have to do to go through graduate programs. And if you find if your goal, is to make a living, maybe it's easier to do something else.
Sure.
Every kid is a natural scientist, right? Humans are incredibly curious. Yes. And then it gets beaten out of you.
I think school is part of that. Because
stop asking that question.
They make them memorize things rather than learn the discovery that.
Right.
That
I mean, in science, in 8th grade or high school science is, here's the Krebs cycle. It was always figured out, memorize it. It's like, well, that's really boring, because if I need to know the Krebs cycle, I can look it up. Science is about the question you don't know the answer to.
Who discovered it and how, right? How did they pick apart the Krebs cycle? That would be fascinating.
But, the science is taught the wrong way. So I think we take that natural curiosity and we kind of beat it out of kids, unfortunately. So,
I totally agree with that, All right, one more question. Did we learn anything from your study of 1918 pandemic virus that was applied to COVID pandemic, in your opinion?
That's a difficult question. I think certainly the lessons of 1918 in general, the public health response in 1918, over and above, just say the sequence of the virus or pathogenicity studies, certainly impacted our planning for COVID and pandemic planning in general. I think that the role of this sort of toxic or pathogenic inflammatory response that we saw in 1918 and that we see in COVID is fascinating. There are lots of interesting kind of molecular pathologic details that, I wish we had time to talk about, in which there are so many similarities between what you see in the lungs of autopsies of 1918 victims and what you see in the lungs of autopsies of COVID victims that, that share, but that you don't see in other flu autopsies, seasonal or other pandemics, 2009, 57, or 68. So I do think that we will learn that there are commonalities in the way that the inflammatory response is shaped. There are commonalities in the way that it leads to induction of pulmonary thrombosis and a prothrombotic environment. In 1918, the lungs were full of blood clots. It was reported in 1918 as a really unique feature. This was kind of rediscovered as a really unique feature of SARS-CoV-2, and yet the pathologists in 1918 showed the same thing. We're right now looking at the molecular mechanisms. It could be that that's a secondary response to a completely different mechanism, an endpoint. But it could be that they share a mechanism. And if they do, then that gives us clues about therapeutic development that might be bigger than a certain class of virus, maybe some kind of host inflammatory mitigator. So these are the kinds of things that I have hope in and that keep us working every day.
Well, let's have you back at some point in the future, and we'll talk about those specific mechanisms, because that would be fun. I think that one of the biggest lessons is that in 1918, we had nothing except public health, and the pandemic ended at some point, right? And here we have much more. So people despair that this will go on forever. I said, no, we can stop it. It just takes time. But we have way more than we did back then.
Absolutely. Right.
All right, that's a special episode of TWiV. You can find the show notes at microbe.tv/TWiV. You can send your questions or comments to TWiV at microbe.tv. If you enjoy what we do, explaining science, please consider supporting us. Go to microbe.tv/contribute. My guest today from the National Institutes of Health, Jeff Taubenberger. Thanks so much. I appreciate your time.
Thank you. It's been a really good conversation. Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.